So welcome to the third video in this short series of videos on how to use PyMOL. We've already discussed why we use PyMOL, what the PDB is and the methods used to generate these files. We've also gone through a quick overview of the PDB. Now I've opened up that file in PyMOL. So let me talk you through the sort of user interface. This here is obviously your visualization window, and this is where you can manipulate the protein. Up here is a command line. You won't need to use the command line in this when you use PyMOL, but it does give you some useful information. Here's the control panel, and I'll remind you, up here is a load of different drop down menus with functions which you'll need to use, particularly this wizard one. Before you do anything, I would always recommend displaying the sequence, and this just makes it much easier. So what are the controls? How do we move stuff around? The first thing you're going to need is a 3-button mouse. The 3 buttons is normally the right click, left click, and the scroll wheel middle click. So it's not recommended you use a trackpad. Left click and hold will allow you to drag the structure around. Right click and hold will zoom in and out. Middle click and hold will allow you to move the structure around like this. Scrolling will actually use, it's sort of like a focus view. It's called clipping, and it allows you to zoom into, sometimes you have quite large protein complexes, and if you want to look right in the middle, this will allow you to hide bits of the structure so you can look right down in the middle. That's the basic controls in this window here. Over here you have this control panel. This control panel has either objects, so like this is an object, or selections. That is a selection. This is a selection. Anything in brackets is a selection. Anything without brackets is an object. So let me demonstrate how to use the selection and objects by modifying this. This is HDAC3 Smart. There's one HDAC here with Smart here, and there's another HDAC here with Smart here. They're the same. So I'm going to get rid of one. How I'm going to do that is I'm going to select one part of the protein. So you can see as I'm selecting it along here, it's selecting it in the window. And I'm going to go over to the selection, actions, and I'm going to remove the atoms. I don't need them. I'm also going to remove these and go action, remove atoms. And I'm also going to remove these. What you do see is this thing here I'm going to remove as well. What you do see is these loads of dots. They're waters. So I'm actually just going to go remove waters. I don't need them. I'm just going to center. So that's how I was using selection to remove part of the structure. Now, as I said before, this is actually 2 proteins, and you can indicate by the 2 chains. So I'm actually going to save this selection. So I'm going to go action, rename selection. And I'm going to call it Smart. Now, if I was to make a new selection, this thing would appear. If I want to get back to the Smart, I'll just click that, and now I've got that selection saved. Now, this allows you to toggle on and off the selection or objects. While I've got this selection active, so it's in the light grey, I can change, I can edit the selection, and it will save that edit. So I'm going to do. I'm going to make this also a new selection. So now I have 2 selections: Smart, HDAC3. Now there's also this molecule, which you can see here. Now I don't want this to be a saved selection. I want this to be a new object. So I've selected it. I can either select it here or I can just select it here. I'm going to go to Actions, Extract Object. I'm going to go Actions, Rename Object. So now this is a unique object separate to these. Okay, so that's the basic principle of selections and objects. Now onto A. You would have seen me use this a few times. This stands for action. So things like deleting, extracting, finding polar contacts, generating electrostatic interactions, aligning. You can align two molecules together. Now the one along is show. This is where we can show different views of the protein. For example, I may want to show this as a surface. And then you can see now it's shown as a surface. But important to note, when you show stuff as a surface, it doesn't hide what was there already. So if I was to do this focus view again, and you can see that it's still showing what this is known as a cartoon view before it. So if I want to hide the cartoon view or hide the surface, I go across to the next window tool, which is hide. So I can hide surface. Cartoon is a really common way of visualizing proteins. It's a visual representation of the backbone, and this is an alpha, this is a helix, and this is a beta sheet. But it doesn't give you any information about the side chains or even the actual backbone. And to do that, you can just go show something like sticks, and that will represent all of the side chains and backbone. Next one along is label. We won't need to use the label, but if you would like to use the label, if you want to label atoms or anything, you can just make a selection and then go to selection, label, click on one of these, and it will show you the label. The last one is color. Color is actually really quite useful because it easily indicates the difference between different parts of the protein. For example, if I ask to color by chain, it is now shown haystack 3 and smart here. So it's really easy to show the difference. That's the basic overview of the layout, the user interface. In the next video, we're going to talk about how to actually use some of the tools to give you some information.